All right. I see that everyone is starting to log in and join us this afternoon. I wanted to welcome everybody to this wonderful mild TBI webinar that we have planned for you. And I'm very excited because we have Dr. Benson back with us, and we also have Caroline, who's going to be helping moderate. Later, you're going to meet some of my amazing partners over at HMR, Fox AE, On Point Legal Nurse Consulting, as well as Strategic Relationships. These are incredible companies that we work with amazing people. And I just wanted to thank everybody so much for supporting these companies, because in return, you guys are helping support us here at Connectionology so we can continue doing all these webinars for you guys. If you have any questions, please put those in the Q&A box. We're going to answer a lot of your questions towards the end of the webinar today because we have a lot that we're going to discuss. We might even go over a little bit, but we hope that you guys will hang in there with us. So I'm going to go ahead now and just introduce our incredible moderator who's joined us today. Caroline, she practiced personal injury as well as wrongful death and product liability. She has extensive experience handling a wide variety of lawsuits that involve pedestrian injuries, motorcycle crash injuries, traumatic brain premises liability, and motor vehicle collisions, where she has successfully obtained millions of dollars in settlements and verdicts for her client. And she has a major passion when it comes for medicine and helping people, and that allows her to maximize the amounts that she's recovered for her clients who have been hurt or even killed. And one of the firm's attorneys, or as one of them, she also takes a lot of time to understand your case and cases that she works on for her clients. She provides a lot of that personal communication, as well as ensuring that everyone has been represented well. So we are just thrilled to have Caroline with us. I'm going to go ahead and hand this webinar over to her, and I hope you guys have a wonderful webinar.
Wow, Ginger, you are too kind. Thank you. I am so excited to be here with everyone today on such an important topic that really all personal injury lawyers need to be aware of and need to continue learning more about, because there's constantly changes and advances being made in this field. And we are so grateful to be here with Dr. Benson. He is a globally regarded behavioral neurologist and imaging neuroscientist who is recognized for his advances in the diagnosis and treatment of brain disorders. During fellowship at Mass General Hospital in the 1990s, he pioneered the use of fMRI to map language in neurosurgical patients with tumors and epilepsy. At the faculty of Wayne State University in the 2000s, his research using electrical and magnetic stimulation of the brain in hemiparetic and aphasic stroke demonstrated proof of concept for the ability to induce neuroplasticity. More recently, Dr. Benson's clinical practice and research has focused on more precise diagnosis in head-injured patients. He's leveraging newer functional neuroimaging, neurosensory, and pituitary evaluations, culminating in more effective treatments and improved outcomes for patients. Dr. Benson's research and clinical practice through the Center for Neurologic Studies are strongly focused on disorders of the brain and cognition. And his commitment to finding novel ways to enhance recovery and function after stroke led to multiple foundation and industry-sponsored investigations into the use of targeted electrical magnetic stimulation of the brain in motor and language impairment following stroke. His research on brain disorders is high impact and cutting edge. And Dr. Benson, I'm going to hand it off to you. Thank you for being here today.
Hey, Carolyn, thanks so much for that wonderful introduction. So yeah, it's really a pleasure to be able to talk to this group today. Yeah, there's a lot that I need to convey to you. And I think it's largely game-changing information regarding objectifying these injuries. When I say these injuries, I mean mild TBI. Severe TBI, it's not hard to evidence. Standard imaging techniques are probably good enough in most cases, but it's the mild TBIs that are really the challenge. And so right around 2002, 2003, I realized that we needed more. And so I kind of made that my main goal to develop advanced imaging techniques. I had cut my teeth on functional MRI back in the 90s, but then I heard about this technique called diffusion tensor MRI that was being used in Italy for multiple sclerosis. And it just seemed to me to be an ideal fit for the white matter injuries that we see in traumatic brain injury. Oh, by the way, I run the Center for Neurologic Studies. I moved my entire operation from the Detroit area to the Tampa Bay area. And I'm speaking to you from St. Petersburg. Yes, I did get flooded, and yes, I'm in an Airbnb now while my house is being put back together, but all is good. And you know, I give a talk on mold and the brain, and so it's kind of ironic that I bought a house on the water that was flooded, and of course I got my ass out of there as soon as I could. Okay, so let's go to the talk. Oops. Okay, so advanced MRI imaging of mild TBI, uncovering the markers of mild traumatic brain injury. So this is the key. We're looking for markers. We're looking for indicators, objective evidence that can reveal these injuries so that we are, in a good position. So you're in a good position to make a case that somebody has more than just psychological problems like PTSD or depression, because we know that's what the other side would like to say. They'd like to avoid conceding the existence of a brain injury at all costs, because that gets expensive for these insurance companies. So yeah, so speaking of real estate, yeah, it's location, 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 and it's all contained in the skull. It's amazing that that there's so much to the brain, and it's all contained within this hard cover, this protective cover that unfortunately can only do so much. So the brain actually sits inside this lower portion of the skull. The temporal lobe would sit here and here, frontal lobe here. Carolyn, can you see my cursor moving? Good.
Yes.
Okay. All right. So the frontal lobes sit on top of this so-called cribriform plate. And a lot of times with coup contra coup injuries, we see hemorrhages in the frontal lobe. This is a hard surface right here. Sometimes the olfactory bulb can get torn. The olfactory fibers run through here and can get twisted and damaged, and people will lose their sense of smell, like my patient yesterday that I testified for in trial. And she had so much going on. It was really an afterthought. And yet her husband said, well, she doesn't complain about my smelly feet anymore. Yeah, she doesn't smell, and she doesn't really taste the way she used to. Okay, so yeah, so the brain is really pretty malleable, pretty gelatinous. I read once that it's got the consistency of a thick oatmeal. So what that means is that the brain can deform. It can swirl around when the head gets knocked. You know, if there's acceleration or deceleration, the brain is moving around. It's got a limited protective shield, and that would be the skull, and to some extent some fat layers underneath the skull and scalp, and then spinal fluid. But the brain is sort of floating in this spinal fluid that surrounds it, including the brainstem and the cerebellum. So for those of you who haven't been exposed to what the brain looks like, there's a frontal lobe, which is really about 2/3 of the cerebral hemisphere volume. This is not really an accurate depiction. The parietal lobe is behind it, the occipital lobe behind that. So if you fall back, you're going to bang the occipital lobe. The temporal lobe right here, cerebellum, brainstem. Okay. Now, deeper in the brain lives phylogenetically or evolutionarily the older brain. So lower vertebrates, lower animals don't have as much cerebral cortex as we do. And they kind of live off of their thalamus and their limbic system, which is highly tuned towards emotional processing as well as memory, largely spatial memory or sensory memory. Okay, two categories of pathology, of traumatic brain pathology, generally speaking. One we call focal injury or regional injury. We're talking about blunt force impact to the head that results in typically bruising or hemorrhaging on the surface of the brain where the impact is, or maybe also, in addition to the coup, there could be the contra coup, which tends to be on the opposite side. Whoops, how did I do that? Yeah. So what happens is with impact, the brain kind of compacts itself, pulls away from the back portion of the inner table of the skull, and then it rebounds and slaps, causing what's called a contra coup injury, which tends to be on the opposite part of the brain. Sometimes we see those. There are other kinds of focal lesions, including bleeds inside the brain, but those tend to be very, very regional, as opposed to diffuse injury, which really accounts for the lion's share of the pathology from closed head injury. Almost everybody with a closed head injury has some degree of diffuse injury, and we'll see why that is. So this diffuse injury results from inertial forces as a result of rapid head, typically rotational motions, linear as well, but rotational seems to be more the primary injury mechanism, and it deforms the white matter. We're going to see more about that. All right, so you can have coup contra coup with lateral impacts as well. All right, so we talked about rotational acceleration. We're talking about the brain rotating, literally rotating to some degree. And I'm going to show you a video that demonstrates that. So MRI, I like to say MRI is what do I like to say about MRI? MRI is a platform. It's a platform that allows for multiple different kinds of scans or scanning. And each one of these different kinds of scans, maybe it's gradient echo or SWI or DWI or FLAIR. These are all different kinds of scans. They are differentially sensitive to different kinds of pathology, or you might say consequences, damage caused by head trauma. So you can see hemorrhages. You can have blocked vessels that cause stroke or ischemia, axonal injury to the white matter, et cetera, et cetera. And then finally, in the chronic stages, you actually see atrophy. There is loss of axons. We're going to talk about what an axon is. And ultimately, the brain substance shrinks, and that's measurable with a type of MRI called volumetric MRI. Okay. So, yeah, so I like to call MRI a one-stop shop. Okay, it fits. So here's one type of MRI. So these images are all acquired in the same patient. Okay, I believe this was an 11-year-old who fell down a canyon, and his parents unfortunately perished. He lived, but he's severely injured. So his structural MRI looked pretty good, but then we saw with the iron sensitive scan, SWI, he had a bleed in his thalamus. We did blood flow scanning to see whether he had strokes and just to see generally where he had significant damage to the brain. We did functional MRI on him in response to language stimuli, and he activated his brain in language areas. We did something called diffusion tensor imaging, which allowed us to map the white matter fibers of his brain, which are essentially the cables or the wires of the brain that connect up neurons to each other. And then we did some biochemical imaging called spectroscopy, just to sort of motivate the idea that MRI is truly a very diverse, versatile kind of imaging platform. What I'm going to talk about today are really the workhorse scans for me. And they include FLAIR, SWI, DTI, and then volumetric imaging. With these, I get the most information in the shortest period of time. These are robust imaging methods, meaning that it's easy to get good data. It's not easy to get good metabolic data or perfusion data. It really requires a lot of cooperation from the patient. Okay, one of the points I like to make to really all audiences is that TBI is complicated and it evolves over time. This is important because there used to be this narrative, and I guess it still exists, that the symptoms are at their worst immediately after the injury. Now, of course, if somebody loses consciousness, you can say, well, that's the worst, right? And then they get better. But not really. Actually, studies have shown in football players, NFL players, that the guys that lose consciousness on the field actually have a better prognosis than those guys that don't. And we think that that's because a loss of consciousness can be an electrical phenomenon, not necessarily an injury phenomenon. With that being said, we know that these biochemical cascades evolve over time, and symptoms evolve over time. I see people with inner ear symptoms from damage to inner ear structures that evolve over a month and sometimes even longer. But long-term clinical outcome is driven by diffuse injury, not focal injury. So by and large, people that have coup contra coup injuries and not much else, they tend to do pretty well. It's the people that have these closed head injuries and maybe there's loss of consciousness, maybe there's a long period of post-traumatic amnesia, these people generally have worse diffuse injury. We're going to talk about that. Okay, now a little review of neuron anatomy. So this is a neuron right here. Here's the cell body. Here's the nucleus. These are the dendrites, which are largely like the antenna of the neuron. Other neurons will synapse on these dendrites. The neuron gives rise to this long axon. So this is an axon, and it has what's called a myelin sheath on it, which may be thick, it may be thin, depending on what kind of neuron or what the function is. Generally speaking, the thicker the myelin, the faster the conduction. Think of the myelin as being like the insulation on a wire, conserving potential changes down the axon with little dissipation of current. All right, so you see then there's the synapse. The synapse is on the postsynaptic neuron right here, etc. Okay. All right. Now let's look at a cross section of a brain. All right, and the gray matter is darker, kind of a yellow color in this slide. And you see the folds. These are called gyri, and these invaginations are called sulci, right? And you know, the more of this you have, the more surface area you have, the more neurons, right? So if you look at a human brain compared with, say, a primate brain, or I don't know, a zebra brain, you're going to see a whole lot more folding. It's almost like we've outgrown the capacity of our skull, so that the brain had to grow in on itself to accommodate the increased number of neurons and increased connectivity between and among these neurons. Okay, so the white matter is here, the gray matter is here. We know, let's see what my next slide is. Yeah, so the cell body of the neuron lives, it's a lot smaller than this, but it lives in the gray matter and gives rise to the axon, which forms the white matter. And the reason it's white is because of the myelin. Myelin looks white under a microscope. Okay, now this is an actual photograph of a brain with the top half of the brain removed, so that you can look at this corpus callosum, which is the bridge between one hemisphere and the other hemisphere. These are not individual neurons or axons here, but these are bundles of axons called fasciculi. And you can see that these axons go across to the other side and they synapse pretty much homotopically, meaning the same area, almost a mirror image. So that ends up wiring up the two hemispheres together. It's more efficient that way. And then here would be a photomicrograph of axon bundles with individual axons contained. You see, and this is important for our later discussion, that these axons run in sheets. They run in parallel sheets under normal circumstances. So let's talk about our first imaging technique called FLAIR, or fluid attenuated inversion recovery. This is a technique, this is a scan that's done on every single MRI scan. It's a structural scan. It's basically a lot like a T2 weighted scan, but it causes the spinal fluid to turn dark. And that allows us to be able to see hyperintensities or scars along the ventricle. This is fluid-filled cavity in the center of the brain. And then we see here, we've got some hyperintense. This is abnormal. It's not necessarily abnormal in an old person, but it certainly would be abnormal, let's say, in a young person. Now, this person was 62, and what I'm trying to show here is this person had deep and subcortical hyperintensities. This would be deep because it's closer to the ventricle. This is subcortical because it's immediately beneath the fold of the cortex, the so-called gyrus right here. Turns out that even though they're both white, they have somewhat different significance or value in terms of TBI. We'll talk about that. So now I'm going to let this kind of talk for itself a couple times. So this is an experiment that was done on an individual who was moving their head back and forth inside an MRI. And what you're seeing is that once the head comes to a stop, the brain is still swirling within the skull. Okay, now I assure you this patient did not come out of the MRI injured. This person was simply moving their head back and forth in tune with a metronome, and then the scanner was obtaining images also timed to the metronome. So this allowed for some averaging to occur. So the importance here is that already you're seeing that there's brain motion that is separate from the bulk motion of the head. That's really important. Now, an investigator at Mass General who I knew while I was there, he's a brilliant mathematician and radiologist by the name of Van Wedeen, did some fancy math on the same kind of experiment. Somebody's moving their head back and forth, and what you see is, remember, the gray matter lives on the outer surface of the brain. You see where these dots are moving. They're moving counterclockwise. But if you look deep into the white matter, you see that the dots are moving in a clockwise direction. Now, remember, the cell body is connected to the axon, right? So it's almost as if your head is going in one direction, your body is going the other direction, and that's not good for your neck, right? That's going to tug on the neck. Well, that is precisely what we see. So here is an image obtained from an old friend of mine who was a former NFL quarterback, who unfortunately had his share of head trauma. And when I looked at his brain, I saw lots. This is one slice now. There was more than one slice to his brain, but he had these hyper intense scars, virtually exactly where these axons and neurons would join, the so-called subcortical white matter, right? So what's happening here clearly is that there was shear or strain that occurred because of the differential swirling of the gray matter and the white matter, resulting in these initially swelling and damage to the axons and then scars. So I published a paper, my group published a paper back in 2017, where we really explored the specificity of these subcortical white matter lesions. They are lesions, they're abnormalities, which I have to tell you, the radiologists on the other side would always say are nonspecific and that they are due to aging or microvascular disease like hypertension. So I ran a study involving 94 controls, 180 TBIs, mostly mild TBI. And what I saw in the controls less than 44 had almost no subcorticals. But the patients, the mild TBI, younger cohort had a much higher rate of subcortical hyperintensities. All right, this was true in the older group. Now, the older group, their baseline prevalence for subcortical hyperintensities was higher than the younger group, as you might expect. right? Due to aging and hypertension, diabetes, etc. But 42% occurrence for the mild TBI folks, huge difference, really in both age groups. All right, so the idea, of course, the take-home message is that I don't let the radiologists on the other side ascribe these subcortical hyperintensities to effectively just aging and other non-specific things, because they always do. All right, so we can cite that paper for sure. Now, what about susceptibility weighted imaging? This was something that Dr. Hakey, my colleague in Michigan, developed back around 2000. It's kind of like gradient echo on steroids. It's much more sensitive to hemorrhage, and not just acute hemorrhage. I'm talking about old hemorrhages, because the iron stays around in the tissue even after the fluid is reabsorbed. So what are some hemorrhages? Well, contusions, like I said before, at the base of the frontal lobe, you have these coup contusions because the base of the frontal lobe is very rough, and the frontal lobe can slap against it. But you also have these diffuse vascular petechiae that we see in the more severe of the diffuse injuries. This is the corpus callosum right here. What's going on here? Well, to keep it simple, normal tissue has a certain propensity to be attracted to a magnetic field, but iron is much different. So because iron has a very high magnetic susceptibility, the MRI signal gets wasted. You lose signal really quick because of the inhomogeneity between the iron deposition and the surrounding tissue. Yeah, and susceptibility weighted imaging developed by Mark Hakey is about 3 to 6 times more sensitive to hemorrhage than gradient echo. And Karen Tong at Loma Linda showed this in 2002 in a paper. So just to give you an example, just to show you how dramatic the difference is between a standard T1 weighted MRI, where things look pretty normal right here, except maybe for this little thing right here. But here's SWI of the same slice of tissue, and every single one of these dark, or what we call black holes, there's a big one right here. These are hemorrhages. Now, this person has a condition called cerebral amyloid angiopathy. This is not a TBI case. This is a TBI case that we published, I don't know, back in 2010 maybe, where we showed a hemorrhage, this type of spherical hemorrhage, but we also see these medullary linear hemorrhages frequently in severe cases. Right, these are all hemorrhages here. This is a TBI. So now let's talk about diffusion tensor imaging. So now we're talking about, we're talking about shear injury. We're talking about deformation of the white matter of the brain. So you can imagine an axon that gets sort of torqued, twisted, over time. It may actually segment and come apart. This broken axon business really only happens in terminal cases where you actually tear the axon and break it like that. The forces to do that, I'm told, are great enough that you won't see this situation in people that survive. On the other hand, what we know, and this is an experiment that was done at the University of Pennsylvania, if you take an axon and you stretch it, you pull it apart, as David Meany did experimentally, well, a few seconds after you do that, the axon kind of folds up like this. This is a reversible state. If you keep tugging on that axon for a longer period of time, what happens is the membranes start to fracture or break, and sodium and calcium and water flood into these broken membranes and cause swellings, these sausage-shaped swellings in the axons. You can see under a microscope. And then days to weeks later, these axons start to break apart, and you'll see these so-called retraction balls, right? So that's a far cry from, you know, a nice linear axon. So the morphology changes. Now, fortunately for us, DTI is able to see, if you will, water flow or water diffusion inside the axon. And in the normal state, the water molecules, these H2O molecules, they're delimited. They can't go across the membrane, so they can go forward, backward, but their flow is largely linear, as opposed to, let's say, water in a glass of water, where the molecules can diffuse equally in all directions. Now, after trauma, the water now, because of these pores in the membranes, the water is sort of intermediate between anisotropic flow and isotropic flow, just to throw those terms out. DTI can see this. DTI is able to measure the water flow in 3 dimensions. And as such, we can use a mathematical representation, or we can use an ellipse to depict the degree of asymmetry, or anisotropy is the term, of the water flow. So here's the normal state. You can see this vector is much bigger than these two, right? So that's your 3 dimensions. Early post-injury, it's, you know, a little bit more equalized. And then late post-injury, you know, certainly in the retraction balls, but also when you lose axons, basically you get free water, and free water is freely diffusible in all directions. So what this allows you to do is to make maps of the white matter of the brain. So let's start with DTI. This is standard DTI. So you can see that the direction of flow is encoded by color. All right, so forward and backward is blue, right, left is sort of purple, and then there's a depth component that looks like it's probably green. Well, there are a couple of other techniques, and sometimes the experts on the other side, if they're MRI jocks, so to speak, they'll say, well, Benson's using DTI, he should be using DSI or DKI or Hardy or one of these other newer methods. And while these methods do have advantages, they take longer to acquire. They require a bit more software. And they really only buy you a little bit more specificity in these crossing fiber areas, where you have multiple fibers going in different directions that are kind of overlapping each other. Otherwise, there's no real clear benefit. So we use this fractional anisotropy to measure the degree of directionality of flow, where FA ranges between 0 and 1. The higher the FA, or the higher the fractional anisotropy, the healthier the tissue. The lower the value, the more damaged it is. So I did a study back in 2007. This was really my first foray into DTI. And I scanned a bunch of controls and a bunch of patients. And all I did was average the FA value over the entire volume of white matter in an individual's brain. So each one of these is a person or a person's brain with an average FA value. Well, what you see clearly is that there's no overlap between the TBI folks who had lower FAs and the controls that had higher FAs. So that was significant. We did have 6 mild TBIs in this group. They were over here, so they were close to the controls, but they still didn't overlap. And what's more, what we found was that the average FA value correlated with injury severity. So on the y-axis, we have the duration of post-traumatic amnesia as an index of severity. And over here, we had the FA mean. And we had this incredible correlation between the DTI index and the severity index. So from there, we were really off and running. We knew we had something that correlated with the amount of injury. Oh, and then just to sort of, I guess, make sure that we had some real tissue to correlate with, because we weren't going to do autopsies or biopsies on people with concussions. We used animals, we used rats, and we looked at 24 rats. And so this is one example of a corpus callosum in a rat, where these red globs are injured axons. Effectively, you can see that this side is more injured than this side. Well, when we looked at the DTI, we found same thing, right? And even the SWI, surprisingly, because the SWI also reflects oxygen extraction from the tissue. What we saw is that this most severe part of the corpus callosum, most severely injured, correlated even with the metabolic dysfunction revealed by SWI.
Dr. Benson, I'm so sorry to interrupt you. This is fascinating, but may we take a moment to talk with our sponsors?
Sure.
Fantastic. Thank you so much for this. I'm already getting great feedback already, Dr. Benson. And there's a lot of questions building up. So when you finish this presentation, we'll be able to address all of those.
Good.
I'm going to bring in real quickly just 2 of our great partners that we have. One of them is Patrick. He's been a nurse in the industry over 20 years. He works with On Point Legal Nurse Consulting, which is a great company that we've been working with, that the Romano Law Group loves working with as well. And Patrick is going to share with you guys some of the great things that they do over there. I will say John Romano raves about those pain and suffering reports. So keep an eye on those. Hi, Patrick.
Hi, Ginger. How are you?
Good. Thank you.
Well, thank you again for having me. And like Ginger said, my name is Patrick Stonich, and I am one of the program directors over here at On Point Legal Consulting and Expert Witness. We've been doing this type of work, so litigation support, for about 25 years. I've been a nurse for about 20 years, and my background started at Thomas Jefferson University in Philadelphia in cardiac ICU, and then went out into community health, the strange world of community health, of home health and hospice. So some of the things that we do over here at On Point We like to start with our attorneys as early as possible with getting cases as they come in on intake. So the sooner we're involved to go through merit screening, organization of your medical records, putting chronologies together, that gets us through the process up to some of our pain and suffering reports, which are non-economic damages reports. They're big reports for TBIs. We do have 3 licensed life care planners on staff that'll be able to put together life care plans. And then one of the big important things that we have as well, besides our wonderful experts, our nursing experts and medical experts, is we have a case management department. So all of the individuals that we're following in the community around the country that have traumatic brain injuries and need follow-up for nurse case managers to make sure the plan is being implemented properly, they're getting the things that they need, and they are making sure that their equipment is working. We have nurses all around the country that continue to follow those clients and report back to the attorneys or report back to special needs trusts, whoever needs to be involved in that whole entire process. So I love being a nurse. I love to teach. I love to talk about nursing. We're here to help. The easiest way to get in touch with us is to send either myself or my coworkers a message. We'll get back to you right away, help with a quick question so you don't go too far down a rabbit hole of trying to figure out some medical stuff. The pain and suffering reports that Mr. Romano always talks about, they're wonderful reports. They're very comprehensive and they really just show a different angle of how a case is, how someone is suffering after an injury. So it's a little bit different. The nurses will do interviews if we can with the client and the family to see how an injury is actually affecting. And today we've decided to, since Connectionology has been so wonderful to us for many, many years, anyone coming to OnPoint will have an ongoing 10% discount for all of our services. So that discount will come off the invoice, whether it's an expert invoice, whether it's a consulting invoice, automatically. If you mention, I think Ginger's name, then we'll know to give you that 10% off. Or you can mention Blue's name and we'll give you 10% off. So thank you. I'll put all my information in the chat. If you need anything, just let me know. I'm here to help.
That is so awesome, Patrick. I will make sure that I send those details again to everyone who's watching today, but what an amazing thing to offer everybody here at Connectionology. Thank you very much. And speaking of how you enjoy teaching, I want everybody to know that Patrick will be teaching next week because we have a webinar where he'll be featured along with Eric Hertz out of Atlanta, Georgia. And they will be doing a nursing home abuse webinar that you do not want to miss. Even if you don't even handle these cases, this is just good information to know because a lot of us have older parents or we will at some point, and you never know if we know somebody who's going through these type of things. So please tune in for that and share it with your friends. But thank you for all that you do, Patrick.
You're welcome, Ginger. Thank you.
All right. Put your information in the chat box. And thank you again, everybody, for staying in touch with him. And speaking of dear friends, look who's next. We've got Blue coming in from Scottsdale, Arizona. It's probably beautiful out there today, but we're excited to have you with us, Blue, because you work with a company called HMR, who's also been with us for many years now. And I cannot imagine doing these webinars without you guys. You also help a lot of our attorneys around the country, and you're working in almost in every single state. So I'm excited to hear what you're going to share with everybody today.
Ginger, thank you so much for the introduction. Patrick, great hair today, man. I gotta say, it's definitely on point, as they say.
Good one.
Anyways,
Nice to see you.
good to see you, Carolyn, Dr. Benson. Amazing webinar you're putting on today. I love the visuals. And I'm just going to jump into it. So who is HMR? So HMR Servicing, we are the premier funding source for personal injury plaintiff law firms nationwide, like Ginger had mentioned. We kind of specialize in catastrophic injury cases. So traumatic brain injuries, spinal cord damage, amputees, the full gamut, right? There's 3 funding buckets, as I like to call them, that we do offer for attorneys and your clients as well. And I'm just going to run through those briefly. First of which is case cost funding. So experts, life care planners, on-point legal nursing, you know, anyone that you need to bring in to shore up your case, depending on your caseload, your litigation load specifically, and how many you have in the firm, those costs can get pretty expensive, pretty astronomical. If that becomes a heavy lift for you, please feel free to reach out. We'd love to step in and help out with that. Also contact me to get more information about any of these. Please feel free to reach out to me directly. Second funding bucket is going to be the medical funding piece. So we operate in 39 states currently for the medical funding. And I always like to mention that just to let you know that we do work with providers nationwide, from Cairo all the way up to neuro and facilities and everything in between. So if that's ever a need, or you have a case outside of your backyard and you don't have providers there, I get that a lot. Please feel free to reach out, bounce it off me if I can help. We'd love to step in. Last is going to be the pre-settlement funding piece. This is a tough time for your client. Maybe they need to pay their mortgage, their child support, their car payment, whatever it is, right? Case by case basis. If that's a need for you, again, please reach out and we'd be more than happy to help. I just want to take a minute to thank you, Ginger and Connectionology. You guys are always so wonderful to us. I love it when we all get together in person and we get to kind of, you know, chuckle and have some good laughs together. So anyways, everyone have a great day. Thanks for having us.
Thank you so much, Blue. We all feel the same way here at Connectionology and love working with you guys, and looking forward to the next webinar. And I believe the coffee giveaway is still coming up. So excited to get that announced. So somebody is going to win 4 bags of freshly roasted coffee at the end of this webinar. And I cannot wait to see who it is. But thank you for all that you do. And please put your information in the chat box for us, okay? All right, we'll see you soon.
Thank you so
Dr.
much,
Benson?
Blue. Yeah, I just wanted to mention briefly, Ginger, I'm sorry to interrupt you, but it's just so important to remember sometimes clients have a hard time, you know, describing what they're going through and their needs. And it's really our job to see, do they need help? Do they need the funding? It can be a very scary thing to be experiencing. And sometimes clients need the help, and it's our responsibility to get that to them timely and not have to wait 2 or 3 years, which is how long these legal cases take.
Totally agree.
No, that's a great point. Thank you for making that, Carolyn. And thanks for having us again, everyone.
Absolutely. All right, Dr. Benson, back to you. We can pull open the slides where we just left off. Can't speak for today, but you guys, thank you so much again.
Maybe we could address one of the questions I saw in the chat. People are asking about these different types of MRIs and the imaging. Dr. Benson, is that something that is just one MRI scan, or could you explain how a patient would get these scans?
Yeah, it's one single scanning session. It's about an hour long. So we acquire all of these scans, meaning FLAIR, SWI, DTI, volumetric, all in one setting. So it's really efficient. Typically, what we do is you send your client to me down in Florida, and the scanner is in Tampa. I'm about 20 minutes from there. I'll spend 3 to 4 hours with your client examining and doing what I need to do. And either before they see me or after they see me, but generally the same day, they have their scan done, and they fly back the next day. That's how it works. Okay. All right. Well, then I'm going to continue. See if I can move this out of the way so I can actually see what this says. Yeah. So, you know, I showed you that graph a little while ago that showed all the TBI dots down below, and the controls were above. There was no overlap, but I knew even then that we got lucky. You're going to have some mild TBI folks that overlap with the controls. It just stands to reason, because we're treating the whole brain, or at least the white matter of the brain, as one thing. And we're generating or calculating an average FA. So what I knew then was that we had to develop some more specific kinds of analysis of the DTI. I had no doubt that the DTI was doing what we needed it to do. It was detecting injury, but we had to start cutting the brain into smaller pieces to be maximally sensitive. So that's what we did. We generated a regional analysis where we cut the white matter into 50 regions. And then the most specific, the finest resolution was at the voxel-based level, or what you might think of as a pixel, but a voxel is nothing more than a three-dimensional pixel. It's a pixel with depth. So that required some image processing to be able to do that, and I'm going to show you. First, let me show you how we look at the global. So these pink triangles or pyramids are controls after we do age correction, which levels out the slope. And then this would be a patient right here. This is a mild TBI. Well, you can see that this mild TBI person has an FA value that's right in line with the controls. In fact, it's above the line. So it's actually not even in the lowest quarter. It's probably up in the highest quarter. Now, does that mean the person doesn't have a TBI? No. What it means is we better have techniques that slice the brain into smaller parts. Otherwise, the injured parts of the brain get diluted out by the normal parts of the brain. So this is what the regional analysis atlas looks like, right? So these are the different regions that we carve the brain into. All right, and then what we do is we plot. We use z-scores where a z-score of 0 is actually the average of the controls. All right, so if you have a region, these are all regions down here. If you end up being identical to the mean of the controls, you're going to be right at the 0 line. Well, you can see this patient had a lot of regions that were below the line. In fact, virtually all of them, only 4 on this plot, were above the line. The others were significantly below the line. This is probably somebody with a pretty bad injury, if I had to guess. All right, and then the voxel-based method I told you about, right? And the way this works is we have to get all of our brains into the same space. We call this standard space or atlas space. We have software that allows us to morph everybody's brain, provided they're not too damaged. And mild TBI folks generally have pretty good looking brains. That allows this really, really robust software to morph the brain. This is a patient's brain right here into standard space. This is the average of 19 control brains. Once we do that, we can then create maps, statistical maps, where we compare each voxel in our patient to, well, back in the old days, 19 controls, right? And just use some kind of statistic to see if there's significant difference in the FA within these many voxels. And ultimately, it allows us to create these nice color maps where we set a threshold statistically. Might be 2 standard deviations, it might be 1 standard deviation reduced, and then we colorize all of the voxels that exceed that statistical threshold. Oh, I didn't mention that that we to create a Z-map, we also have to create a variance map or standard deviation map of each voxel, so that we take into account the fact that normals have variability in their FA. So we don't want to deceive ourselves. We do want to take into account. So that's what the Z-score map does. Okay. And this is what a typical map would look like. We end up typically showing 12 of 100 slices, and essentially just showing those slices that reveal the most abnormality. Okay, and this is slicing the brain from the bottom up to the top of the brain. This is millimeters below the origin. These are millimeters above the origin, etc. Okay, so now let's put this all together. So this is a FLAIR image in a patient. Let's see if I have any. Let's see. A shows the FLAIR image with the ventriculostomy tract. All right, so this was a severe patient with severe injury that apparently had swelling of the brain, so much so that they put a ventriculostomy tube into the spinal, into the ventricle to relieve some of that fluid and to decrease the pressure. But if you look at that image, that's about the only abnormality you see on this structural image. Now we go over to SWI, which shows hemorrhaging or iron deposition, more precisely, from old hemorrhage. And what do we see? Well, not only do we see the ventriculostomy tract in full, but we see hemorrhages. Boom, this is a hemorrhage, this is a hemorrhage. Hemorrhage on the opposite side of the brain, not revealed by the FLAIR image. Here's a deeper hemorrhage. And then DTI, which is going to show you damage to the axons from shear, is showing you even more. It's showing that there's damage, axonal injury in the left temporal lobe, which you don't see on the SWI. You certainly don't see it on the FLAIR. So I think of these as different channels of information. They're pieces of the pathology pie, if you will. And because damage comes in different flavors, we have to use different types of MRI scans to capture as much of the pathology pie as possible. They're complementary. And this would be a typical report form that I would send you. So we talk about the structural imaging along with SWI and gradient echo, and then we go into diffusion tensor imaging, giving global, regional results, voxel-wise results. Now, voxel-wise results are going to end up being a map. Regional results are going to end up being a table and some graphs. So now some examples, some cases. So case ZD, this was a severe TBI. 19-year-old man was in a car accident, skidded on black ice, hit a tree, right? So only one vehicle was involved. I don't know if alcohol was involved, but skidded on black ice. The person was in a coma, right? GCS, Glasgow Coma Score of 3. You get 3 points for showing up. So this person was in a coma. He was in a coma for 2.5 weeks. And we got him 17 months post-injury, right? So long after the injury. And he was in rehab at this point. This is way back at the beginning, 2011. All right, this is what his brain looked like. Here's the FLAIR. Looks pretty good, actually. Maybe a little bit of atrophy for a 19-year-old. Look at the SWI. Just many scattered hemorrhages, linear hemorrhages, round or spherical hemorrhages. Yeah, this is a bad diffuse injury. He's got diffuse axonal injury, as we're going to see, but he also has diffuse vascular injury. This is what a severe would look like. All right, here's his DTI. He's got damage to the brainstem. He's got damage in the corpus callosum. He's got damage all over the place. In fact, the threshold that I used was 3. That's very rigorous. We certainly would see more abnormality if we used a lower threshold statistically, but that just shows you how severely injured this person was. Yeah. Another case, 23-year-old guy working for the Ohio Department of Transportation as a flagman. So he was out in the field on the highway, semi hit his truck going 60, which struck him in the head. Now, he doesn't remember any of this. And I sort of had to piece together what happened. He was in a coma for a week, broken nose, fractured left orbit. So he certainly had a head injury, right knee ACL, pneumothorax, which means that he had air inside his thoracic cavity, outside the lung, because the lung was perforated. Early on, he was slow and disinhibited. That disinhibited means that he was impulsive. He did not inhibit his impulses. Retrograde amnesia for 2 days. Well, oh, that's retrograde. All right. So if you have a retrograde amnesia, that means that you have a loss of memory prior to your impact or injury. Generally, if you have injury or amnesia that predates the injury, that's a more severe injury. I mean, I've got people that have retrograde amnesia for a month, sometimes even longer than that, but that's still pretty significant. And then supposedly had a remarkable recovery, according to his fiance anyway. She was still his fiance, but had residual memory, vision, neck, back, and knee problems. All right, I'm not going to go through all this, but he had all kinds of symptoms. When I saw him, cognitive symptoms, I break symptoms up into cognitive, emotional / psychological symptoms, and physical symptoms. So he had many of each. On examination, so my neurological examination, which is more detailed than most neurologists because I'm a card-carrying behavioral neurologist. I did a fellowship at Mass General, and so when I examined him, he had many deficits, language. He was frontal. He had trouble with abstracting. Had trouble drawing, had motor findings, had gait findings. Neuropsych examination pretty much showed what I found on exam. And then this is what his brain looked like. These are all hemorrhages, right? We've seen these before, linear hemorrhages and more spherical hemorrhages, pretty typical. Here's his DTI. Hemorrhages again in the left hemisphere corresponding to his language deficits. DTI also revealing a predominance of left hemisphere abnormality. Now, DTI is not really good for showing the surface of the brain because of all the folding. But when you see this white matter injury right here, it's a good correlate of the cortical hemorrhaging that he had. Okay, now I want to talk about volumetric imaging. This is the consequence of brain damage and literally loss of brain substance, loss of axons, and to some extent loss of neurons, right? We can measure using computerized methodology. We can measure volumes really up to the level of small nuclei inside the hippocampus and amygdala. It's very detailed. It's very high resolution. It's very sensitive. So just to show you what one of these reports looks like for Volbrain, which is one of the programs that we use. Now, red means that the structure is smaller than it should be. It exceeds 2 standard deviations reduced in size. So here you see this is normalized for brain size, 25%, 25.48% white matter. It should be, under normal circumstances, the 2 standard deviation below the mean would be 27.45 for the controls, 39.74 above, so plus or minus 2 standard. So this person exceeds 2 standard deviations. That means that this person is in the fifth percentile or less. Well, 25 versus 27, I'd say it's probably more like the 3rd percentile compared with the control group. That's rough. Now, interestingly, the gray matter is within normal range. The spinal fluid, on the other hand, is higher than 2 standard deviations above the mean. And the reason for that is that if we lose solid brain substance, either white matter or gray matter, something has to basically fill in the slack, if you will, and that's the spinal fluid. So we see this a lot with white matter injury caused by closed head injury. Okay, and then it gets even more specific. We can look at the right hemisphere, the left hemisphere, etc. Now, one of the nice sort of quick and dirty results that I look at is something called lesion brain, which, yes, it does look at the number of white matter lesions, but I love this series of 3 graphs. It shows you white matter, gray matter, spinal fluid. Here's our patient. Okay, our patient, this particular patient, is about 1 standard deviation below the mean of the controls. There's over 1,100 controls that are looked at here across this broad age range, not 0 to 100, but probably more like 9 to 90, something along those lines. So 1 standard deviation reduced, gray matter, not much change. Remember, TBI primarily affects the white matter, not the gray matter. Spinal fluid, about 1 standard deviation above the white matter. Pretty typical of what I see. Now I'm going to finish with another type of volumetric program that we use, FreeSurfer, which was actually developed at Mass General around the time that I was there by a couple of guys from UC San Diego. And it's pretty nifty. It relies on inflating the brain graphically, and then assigning anatomy based on this inflated brain. But really what we use it for is the anatomic labeling and parcellation. Right, this allows us to be able to create regions, anatomical regions that we then measure the volume of. Well, I should say FreeSurfer does that for us. Okay, and as always, we compare our patient to a number of controls. For FreeSurfer, we use our own controls. We have 50 controls, and we age correct. This allows us then to measure volumes and within certain regions. It also allows us to measure cortical thickness. So this is literally the thickness of the gray matter, right, where the white matter is deeper. Remember, I told you about the folds of the brain. This is important because we have a 5-layered cortex here, actually 7-layered cortex in some regions. And when neurons are lost or white matter is lost, the cortex gets thin. All right, so we have the capability of measuring cortical thickness. And we then graph the results a few different ways. This is cortical white matter. We've got frontal lobe, temporal lobe, parietal lobe, occipital lobe, cingulate. We put our 2 standard deviation lines on here so we can quickly see where we're below the line, where certain regions are exceed 2 standard deviations below the mean of the controls. So we can quickly see where we have abnormalities. Okay, so yeah, that's all I'm showing for volumetric. So now let me try to tie this together. So we have imaging biomarkers of brain injury. Hopefully some of this made sense to you. The real take-home message is you don't have to understand this stuff in great detail. I do. And I am able, with these imaging and measurement techniques, I'm able to validate the existence of a brain injury with these objective biomarkers. I'm not the only one in town doing this. There are a lot more groups that are getting into this now. I was certainly one of the first, and I would say that we're still doing it, I think, at a higher level than most, largely because, as you know, I am a neurologist and a behavioral neurologist. I see the patient, I examine the patient. I'm able to correlate the behavioral findings as well as, we haven't talked about it, but oftentimes your clients will have pituitary hormone issues from a damaged pituitary gland. They might have inner ear injuries. I'm able to pull all of this together. And certainly the imaging allows me to determine when there's a brain injury, or I should say an injury to the brain itself, what type of brain injury it is. I have to tell you, there are times when there's not much in the way of injury specifically to the brain, but we've got somebody with florid pituitary problems, and they're down and out, and they can't work, they can barely function. I did a follow-up with somebody today who's in that boat. So the imaging is helpful for determining who's got a brain injury, but it's also helpful for determining who's got something else. These markers have diagnostic and prognostic ability. Trauma results in multiple pathologic consequences to the brain, which can be detected with specific MRI sequences. I think you get that now. Multimodality MRI is sensitive to even mild TBI, not just moderates and severes. Neuroimaging is objective. I do it the same way every time. I do it blinded, I should say. The imaging is analyzed blinded to everything else about the case. So it's independent. A paucity of imaging findings suggests alternative explanation for symptoms. I mentioned pituitary, inner ear, visual, etc. Okay, now, if you've got a case, and I hope you do or will in the future, and you'd like me to evaluate your client, the way to do that is to get in touch with my CEO, Alina Dalali, who also happens to have her JD. And she is in Boca Raton. Like so many of us, she's working remotely. In fact, I'm the only one in Central Florida from my group. We're all operating remotely, including the rest of my staff that I retained from Michigan, with whom I've been working for over 10 years. Here's her email, here's her phone, and I'm open to any questions that you might have now.
Okay, perfect, Dr. Benson. And I echo all compliments for Alina. She's been fabulous to work with. And I know there's a lot of people who've already been emailing her for a copy of your slides. So please email Alina if you guys would like a copy, and then you can also reach Dr. Benson that way as well. There's some awesome questions that have come in. So we're going to start addressing those. But I just had two more partners I was going to bring in real fast. So that way they can share with you guys all the good stuff that they do. So if you're okay with it, Dr. Benson, I'm going to have them speak just for a minute, and then we'll give everyone else some time to get in their last minute questions for you.
Okay.
This is one of my favorite parts of the webinar, though, is getting to hear what everyone is working on and what they need help with. So I'm looking forward to it. I've got Nathaniel coming in from Fox AE. This is a great company doing all kinds of amazing animations, demonstratives. They can create almost anything, and they work with a lot of great experts in industry to create these images and these videos. And I'm excited to have him here with you guys to tell you all the things that they do over there. Nathaniel, it is great to have you with us.
Thanks once again for having me again. And thanks for taking or letting me have a few moments of your time to explain what we can do for you. So first off, I'm a mechanical engineer. I graduated from the University of Missouri, Columbia with a mechanical engineering degree. We have multiple medical background staff on our team, which we use to create accident reconstruction. We use them to do surgical animations, still shots, just general injuries, pretty much any graphics that you need to visualize for your case, we can probably do for you. But let me show you, excuse me, a quick animation of a brain injury that I believe we didn't work directly with Dr. Benson on this one, but I think we worked through the attorney with him on this one. And so here we take the MRI scans and we reconstructed all of these different, I believe, axonal shearing in this case. And we show before when the incident occurred, and then later on down the road, she had some more scans. So we take each individual scan and reconstruct all the little, in this case, little axonal shearings, which if you look at one scan, it does not look very impactful because you have to look at multiple scans to be able to see the big picture. And so we took this, and this is the initial time of the incident, I believe. And then we slice through the brain to show where exactly those axonal shearing injuries are located in the brain. And so we get into highly detailed animations to make sure we highlight and pinpoint each injury in the brain. And so here, in just a moment, we will switch over to the other set of scans, and you can see how much that injury has grown or has become worse. And so we fade out the brain itself so they can see all the damage inside the brain. And then once again, we do the cuts in the brain just like an MRI scanner does, and shows each of those parts that are damaged. And I'm not totally sure the outcome of this case yet. It still might be ongoing. But you can really take what's considered small by just looking at these scans and turn it into a 3D, easily understandable damaged brain. And so that's just a small portion of what we can do. But I also wanted to take a quick moment to show you our Fox Prime website, where you can go and grab all sorts of different pre-made out-of-the-box injuries, surgeries. So we've got cervical spine. You can come in and get different cervical fractures. You can get herniated discs, or even a complete fusion animation. This one's a still shot, but we do have animations in here as well. So once again, thanks for having me, and I'll put my info in the chat.
Thank you so much, Nathaniel. And that is very cool. I love that website. And I know you guys keep adding more things to it. But I sure hope that everyone will reach out to you if they have any questions. But we love working with you guys. And thank you so much for all that you do for us.
Yep, thank you.
Nathaniel, I do see we have a question for you, and it's a really good one. We all know how difficult and time consuming it can be to obtain medical records, but what types of scans, films would Fox AE need to make that type of video? MRIs, CT scans, or anything else?
Yeah, it depends on the injury, of course. For like fractures and stuff, I believe CTs are the best ones for those. I believe MRIs are probably the best for the brain. We also have utilized DTI scans as well. But yeah, if you are questioning what type of scans we can use, just talk to me and we can figure out what we can do with those scans, and then figure out from there.
Perfect. Thank you so much, Nathaniel. All right, we'll see you back here again very soon. And I know
Sounds
that
good.
Fox AE is going to be with us. So is Caroline. We're all going to Texas in about 2 weeks to the Fort Worth Stockyards for our second annual Depositions, our trial seminar. There are only literally just a couple spaces left because we're only taking a small amount of attorneys with us. So that way you guys can break out into these deep dive discussions every day. I cannot wait. So if you guys would like to come, now's the time before it sells out. So we hope you will check out the agenda at connectionology.com in case you'd like to come. But thank you so much for being there with us too. And now, real quickly, I'm going to go over to Samantha with strategic relationships, and then we will jump into questions. Strategic relationships, we've been working with this amazing company, including Paul, gosh, for many years now. And I am just blown away by what they do, because they actually will go out and find these ex-employees of these companies and literally change cases. Like they will, you might think you have a case worth nothing, and then it turns into like $1 million case because they find somebody who brings forth information that you really need. So much value with this company. Samantha, thank you for being right here for us, and can't wait to hear what else you'd like to share.
Yes, so thank you very much. And we are counting down to going to Fort Worth and meeting everyone in person. What Strategic does is we make it super easy for you to find the information that you need that is going to crack your case wide open. Just from this webinar and the other webinars I've been part of, being an attorney is hard enough. So we make it super easy for you to identify your defendant's former employees. All we need is a defendant's name, the job titles that you think have the information that you need, and a time frame, and we can get you 30 to 40 people that can speak to the processes and give you insider information on companies or if you're looking for witnesses. So this is a very easy process, and we do it pretty fast. You usually get your report within 2 weeks, and it's just under 8,000. So it's not for every case, but for cases that you think this could be really valuable. We also have the option of doing a mini EO, which will be half the names for half the price. So there's, we can work with you and what your case is about. So looking forward to seeing everyone and discussing your cases to see if tapping into former employees is an avenue for you, just another tool in your arsenal.
Absolutely. And I do know that Paul, back in his prior life, he was actually a private investigator for like over 30 years. So he brings forth a lot of this experience. And I think you guys, Samantha, are also doing class action.
Yes, we do everything from mass torts, class actions, employment premises. We've even done some former tenants for some premises cases, and also former coworkers for asbestos. And so this tool can be used in all types of cases.
Okay, awesome. Well, thanks for being here with us.
Thank you.
Samantha, she's already put her information in the chat box. Go ahead and save it, because you never know when you're going to need to call her. But thank you so much for being here with us, and we'll see you soon.
Thank you. Bye.
All right, Carolyn, I'm going to hand it over to you, and let's get some of these questions answered.
All right, we have lots of amazing questions. So Dr. Benson, I will try to get through as many of these as we can. It looks like generally people are wondering, when should these scans be done? Is this something that is ever done in the emergency room? Or if a patient is, let's say, 2 or 3 months out, is that too late? Is a year out from the injury, is that too late? And you have your mute button on.
Okay, so I was just saying that that's a great question. And I didn't really talk about that, but that's the beauty of these techniques, that you don't get them acutely. In fact, insurance won't pay for them acutely in the emergency room. And really any time after the injury. And I would say that a year to 2 years is ideal. The reason for that is that I like to see the patient when they have plateaued or when they've reached MMI, some would say. That way they're not a moving target. I don't need to necessarily examine them again. They've maximized their recovery. And there's no loss in sensitivity of these imaging techniques. The iron stays around. The axons don't grow back. In fact, we tend to see more atrophy, which means the volumetric is even more sensitive the longer you are out from the injury. So my ideal is 2 years or more after the injury. So hopefully that answers the question.
That does. Thank you. And another great question is, it seems like TBI symptoms are similar to injuries to the pituitary gland. How can a lawyer know whether the pituitary gland has been injured?
Well, interestingly enough, we just found out yesterday that we had our paper accepted by the Journal of Neurotrauma on this very subject. So the subject of the paper is differentiating pituitary injury, specifically growth hormone deficiency, from TBI. I'd be happy to give another talk on the topic, but I will say that they are fairly discriminable. As I was talking to this patient today, the follow-up, she's got a huge amount of fatigue. She is now 7 years post-injury, and her fatigue is terrible. Her sleep is awful. She goes to bed around 11:30. Sometimes she doesn't fall asleep till 4:30 in the morning, she tells me. And when she does sleep, it's maybe for an hour at a time. That's sort of classic growth hormone deficiency. And there are many other symptoms that go along with it. So the simple answer is, if your client has a lot of fatigue, and they're more than 1 year out from their injury, there's a good chance they've got pituitary injury, and we need to get them evaluated.
On MRI, are you able to differentiate various findings or artifacts that could be age-related occurring over time compared to those that are directly caused by the traumatic event with reasonable probability? And if so, what are you looking for? That's a great question. Obviously, the defense is always going to say, well, it wasn't the traumatic brain injury, it was age-related, or it was sports many years ago, or it was a hormonal pituitary issue. So what do you look for?
Yeah, and that is another really awesome question. And it's not always easy. I've already told you that those flare abnormalities, those hyperintensities, differentiate, right? So if we've got, as was the case for this patient in trial, she had only subcortical hyperintensity. She had no deep hyperintensities. They were classic TBI. What else? When we analyze the volumetric and the DTI, we correct for age. So we have ways of getting rid of the age effect. That's really important. So yeah, you know, the experts on the other side, so-called experts, are going to throw as much mud as they can to sully the case. But I would say that we have, you know, we have responses, we have answers to them.
A lot of times when we as lawyers work with experts, we want to be getting a lot of the prior medical records and sending them to our expert. Is that something that's important to you? And if so, why?
Well, it's huge. And yeah, and that was a part of the question I didn't answer, the last question. Yeah, past medical history, including prior concussions, are important. Now, so for instance, this case that I testified on, she had an MVA maybe a year before her slip and fall, but the MVA was relatively minor, and she had mostly lower body injuries, and she had largely recovered. So, you know, of course, the past medical history is important, and prior injuries are important. One thing we know is that the greater the number of head injuries or concussions, the more likely it is that you come out worse from the last one. So there's a cumulative effect. There's a concept called cognitive reserve, and you know, the message is that you got a certain amount of resiliency, but like the football players that I've seen, you know, after a certain number of those concussions, you don't bounce back. You know, you get worse and worse and worse. So yeah, of course, past medical, or I should say, the medicals are important. I don't like to be overburdened by irrelevant medical records. So anything that's germane to TBI is what I'm looking for.
Thank you. What about carbon monoxide exposure? Can the testing that you're talking about, can it discover, diagnose brain damage from carbon monoxide exposure?
Great question. I actually have a talk on carbon monoxide because I've actually been doing carbon monoxide for the past several years now, and the answer is yes. The volumetric imaging, which is very precise, can identify volume reductions and cortical thickness reductions caused by carbon monoxide, which has an immediate mechanism of hypoxia or low oxygen. And we know that low oxygen effectively causes damage to the most vulnerable neurons. Those tend to be the deeper neurons of the limbic system, but also the motor cortex and the cerebellum. So yeah, I can help you with those cases.
Dr. Benson, what is the cost? What is the cost of getting these scans done? You mentioned it would be a 1-day process for the patient, and then what would be the cost of having you retained on the case?
Yeah, the upfront cost is somewhere between 10 and 13,000, and that includes the visit with me and reports. That includes the MRI and reports. There are some add-ons like the volumetric, which is an additional 3,500. It's very labor intensive. Then there's a record review and retainer, but Alina can tell you all of that.
Are there any treatments? That's a big one. Let's say the patient has been diagnosed with a mild traumatic brain injury. What then? Is there anything that can be done to help them?
Well, I love that question, and it's not a question that I'm often asked by attorneys, I have to tell you. So I really appreciate that, because I made a deal a long time ago with the attorneys, and that was, I'll see your client for you, but you got to follow my recommendations, which are geared to helping the patient medically, treatment. So there are things that we do that make a huge difference. There's a visual condition called binocular visual dysfunction that we see with almost every single TBI case. I generally will use an optometry group in Michigan, but she's trained people all over the country now. And I think we're up to maybe about 80 different people across the country that use microprisms to recalibrate the eyes with this condition. So that's one treatment that can make a huge difference with dizziness and headaches and visual symptoms, including tracking moving objects. The other big ticket item is this pituitary thing. And I find that probably about 80% of the patients that I see referred by you guys have pituitary hypofunction. Their hormone levels are low. It might be testosterone. It might be human growth hormone. Easily treated with hormone supplementation. The benefits are huge. We see patients with growth hormone deficiency make dramatic recoveries, which is why I hate to miss this. It's so devastating psychologically for people to have it, physically, cognitively, and yet almost everybody turns around with treatment. Other treatments, there are other treatments for pure TBI, something called neurofeedback, which is a type of EEG-based biofeedback, can be very, very useful. And sometimes patients have, as I mentioned, inner ear, vestibular, and auditory issues. And sometimes that is caused by a peripheral problem, meaning inner ear and not brain. And typically the cause of that is that there is a fracture or a break in one of the semicircular canals. Treatment might be a diuretic called Diamox. But if that doesn't work, and we've had some professional athletes undergo surgery because they needed a quicker fix, and that can be a game changer, getting people's balance and dizziness corrected, and even improving hearing.
Very interesting. Is there a blood test that can be done or should be done to look for those hormonal or pituitary issues?
Yeah, unfortunately, they're not easily done. The way that they're done is to do what's called a growth hormone stimulation test. And when that test is done, a number of other pituitary tests are done to assay virtually all of the pituitary hormones and many of the peripheral hormones. So that requires the gold standard test called the insulin hypoglycemia test. So you literally give somebody insulin unless they are a heart failure patient or a bad diabetic, and you drop their blood sugar below 40. It's a stress test for the pituitary. It's really sensitive. And so that's done on one day. And then there's another test that's done on the other day. So that's a two-day visit. Generally speaking, when we see people with pituitary issues, they also have the vision issues. So what we do is we send them to Michigan, where I used to be for over 10 years. And they get the vision test, they get the pituitary test. It's very efficient that way. And they almost always get better from both of those. So it's a win-win. It's also obviously additional lines of evidence supporting the significance of the brain injury. So it really is a win-win.
I love this next question. It is reported that PTSD produces brain volume loss. Can that be differentiated from TBI volume loss? If so, how? And one of the reasons I like this question is I actually had a case where the defense attorneys were trying to say it was my 25-year-old client's anxiety and depression and mental health problems that were the issue, and not the car crash that broke her nose and caused a TBI.
Yeah, so yeah, it's true that probably more severe and more chronic PTSD has been shown to cause atrophy in certain limbic regions of the brain. It's not the easiest thing in the world to see. I don't think that it's as sensitive as I'd like for it to be. It's certainly differentiable from TBI, because those regions that get affected in PTSD are generally not the regions that are affected in TBI.
How about the timing of treatment? I know you said that it's good to wait some time to get the diagnostic radiology done, but how about the timing of treatment? When can that be started?
Yeah, there's no reason to delay treatment. I mean, I'm not saying that you lose anything by waiting a few weeks. It depends on the treatment. Now, when it comes to the pituitary, what we know from studies, published studies, is that lots of people will have pituitary problems in the first year after a TBI, and essentially, if they don't recover after a year, they're probably not going to recover. So insurance doesn't usually pay to get the testing done before 1 year for that reason. And I don't have a problem with that, right? Because if they're still really fatigued and having insomnia, and they've gained 30 pounds and they have zilch motivation after a year, it's not the brain injury anymore, it's the pituitary.
Does an early scan help for comparables? What about pre-existing conditions? And 2 years out is a bit long for most car crash cases.
No, it's really not. Of course, CAT scans are going to be done early, and lots of times people will get standard MRIs done in the first year or so. And that's fine. You know, typically they're normal, they're negative, so they don't really add much to the case. And what I'm saying is a year, 2 years out, if your client is still having problems, chances are we're going to see something on our advanced imaging, and that's going to add value to your case.
Dr. Benson, is there a mechanism whereby lawyers can order these scans to occur at hospitals or local doctors that are more local to the patient, and then forward them to you so you could write up the case?
Another good question. I'm always asked that. And unfortunately, the answer is no. And the reason is that in order to do the imaging the right way, and particularly the DTI and the volumetric imaging, you want your control scans to be done precisely using the same MRI and the same pulse sequence, the same type of scan as the patient. Otherwise, it's very difficult to compare between controls and patient. So the short answer is no. If we're going to do it, we want to do it the right way. We don't want to open ourselves up to, you know, to the defense radiologist who says, oh, you know, Benson didn't know what he was doing. He compared apples to oranges. We don't want to do that.
Are there any new treatments involving the use of magnets?
So we're back to treatment again, huh? Okay. Well, I used to do something called transcranial magnetic stimulation, along with cortical electrical stimulation. And yeah, TMS seems to be pretty good for depression. You know, it's probably even better than electroconvulsive therapy. But apart from that, we don't have any protocols that have been approved, let's say, for TBI. Now, you know, 10 years ago, I would have monthly talks with Jay Maythaler, who headed up physical medicine and rehab. And we really felt like there was a way forward for magnetic stimulation in TBI. But he changed jobs, and, you know, we kind of moved on. It's just not there yet.
Thank you. And this is an interesting question about long COVID. It seems that TBI has some of the same symptoms as long COVID. How do you respond to the argument that the patient has long COVID and not a mild TBI?
Yeah, boy, long COVID seems to interact with everything. All right, how do I answer that question? I could punt that question, but I won't. So look, timing is everything. The symptoms are everything. You know, ideally, there's a real TBI there, and the onset of symptoms coincides with the onset of the TBI, or at least shortly thereafter. Sometimes the cognitive issues aren't recognized early on, at least by the patient. Brain imaging is probably the best discriminator between the two. There are patterns, and I've shown you some of them today, that are highly indicative of TBI. On the other hand, I'm not sure if we know if and what type of patterns long COVID has. In fact, I'm working with a group on mold, and there's some indication that that there's an immune predisposition or a genetic predisposition to long COVID, Lyme disease, and mold, and that about 20% of the population has a genetic predisposition to develop immune abnormalities and chronic inflammation. And what I've seen is that mold in the brain causes increased volumes by virtue of inflammation in these brain regions. So that may be the case with long COVID, but I don't know.
Thank you, Dr. Benson. And we had a question about the Daubert test. I can partially answer that one. The question is, are most courts allowing DTI to get past the Daubert test? The answer is yes. We've had great success having this admissible at trial. It doesn't mean that the defense won't bring the motion, but we've had good success getting past the motion. And of course, you have to work with your expert and get an affidavit, get the deposition testimony sometimes before going to the hearing.
Yeah, and I would echo that. There's only been really 2 or 3 times that I've been limited. And we're talking about probably close to 100 cases in total for me. And I think you're right that you have to work closely with your expert. Those times where it wasn't allowed, I wasn't even told about one of the cases until the 11th hour, and it was really too late. So yeah, I think by and large we're getting it in, and we're winning a whole lot more than we're losing.
Awesome. That's so good to hear. I think we've covered most of the questions.
Yeah, this has been fantastic. And I would definitely encourage you guys, if you have more questions, to reach out to Alina, who can get you in contact with Dr. Benson. And I'm sure he would love to work with you guys and help you out even more. And we are definitely having you back next for the mold webinar. I'm going to make that happen. Thank you.
Have I given a talk on pituitary yet?
Not yet, but that'd be a great one.
I'd like to actually do that one. I'm sort of making it my mission to spread the word on this because it's so common, and yet so few people and so few attorneys, understand it.
Absolutely. Well, if you're okay with it, we're going to line you up for a couple webinars. And I know we would love to bring you in. I'm going to be working with John Romano because our 4th annual TBI seminar is going to be coming out in May 2025. And I'm in the middle of getting the contract set for that venue. We're either going to Nashville or we might choose a new city this time around.
New Orleans.
We'll definitely let you know. That's a great one. Before we head out, Dr. Benson, we brought Blue back in from HMR because I know everyone is anxiously waiting to hear if they've won the coffee giveaway. We've done close to 400 webinars now, different topics, different speakers, and HMR has given away thousands of bags of coffee. So don't feel bad if your name's not called today, you guys, because there's another chance to win next week. We actually have 2 webinars that we'll be doing. So you can go to connectionology.com to sign up for those now, but you'll have many more chances to win just in case. Blue, it's great to have you back with us.
Thanks, Ginger. And I just want to add real quick that Dr. Benson is a perfect example of the kind of expert reports, all the things that you would want to bring in on your case to shore it up and, go do the best you can. And if you need help funding guys like Dr. Benson, I know a guy.
That's you. Well, you guys help with a lot of different things, and traumatic brain injuries is definitely a catastrophic injury that you guys do a lot of work with our attorneys. So thank you, Blue. And we appreciate
Absolutely. All right. Well, let's get to the coffee giveaway. And fair warning, this coffee is it's stout. Okay. It's going to get you going for the day. So be careful with it. Our winner is out of the great state of Texas, Sugarland to be exact, Aurora Martinez with Leon Law Firm. Congratulations.
Awesome. Congratulations to Aurora. All right. I love those Texas winners. Well, that's wonderful to hear, Blue. Both you and I, we can reach out to her after this webinar to make sure that she gets this coffee. And don't worry, guys, there's another chance to win again next week. But thank you, Blue, for coming back here. Thank you for all that you do over there. And we hope to see you back here soon too.
Absolutely. Always a pleasure. Thank you, everyone.
Yeah. And then also, last but not least, I just want to thank Miss Caroline for being here with us today. We will have you back, and I'm looking forward to doing more webinars and then having you also speak on a couple webinars for us too.
Absolutely.
Hey, and last but not least, I'd like to thank Caroline and Ginger for holding just an amazing webinar today. I really enjoyed it, and I look forward to to doing more for you.
I'm looking forward to it too. Thank you so much.
Thank
All
you,
right.
Dr. Benson.
Do either of you have any last minute comments before we head out?
This was wonderful. This was wonderful. And I want to make a quick comment just about a treating provider and the radiology. Remember, radiology is objective. Dr. Benson covered that. But with him as an expert, he's also a treating provider that can get into the symptoms and see a patient, which is so important to us. And it's also one way to be able to defeat the defense radiologist who cannot do that clinical correlation.
Exactly. In fact, just to sort of extend that thought, with regard to the Daubert issue, I'm asked a lot if I agree with RSNA and these other societies' stance about DTI not being generally accepted by the radiology societies. And look, I'm not a radiologist. I've spent my career in research. My training and my expertise goes beyond what radiologists do, as Caroline just said. I'm not beholden to the same gatekeeping mechanisms that the radiologists are. I don't use the radiology in isolation. That's really important. We oftentimes the radiologists want to frame the narrative narrowly. They're wrong, and we shouldn't let them dupe us. So again, remember that I'm not one of them. I am a multi-channel type of treater and expert for you. And I think in terms of value, you're getting a lot. The radiologist would probably charge the same, and all you're getting from them is radiology. You're getting a whole lot more from CNS. You're really getting the whole picture. So I just wanted to add that.
Perfect. Thank you guys again so much. And then keep an eye out for your emails, because I'm going to be sending everyone some follow-up notes from this webinar. And again, I'll make sure that you get Alina's email to stay in touch with Dr. Benson. And if you have any more questions, you can also reach out to Carolyn as well. Thanks again to our amazing partners, and we hope you guys have a wonderful afternoon. We'll see you back here again next week. Bye, guys.
Thanks again.
Thank you.